The next form of cell death that we are going to start with is a very important topic in terms of the MCQs that we have, a very favorite with the examiners amongst all the types of cell deaths that we have. This is known as apoptosis. This is known as apoptosis. Now students, if you just go into a slight historical aspect of this word, this takes its origin from a Greek word which means falling off. What is falling off? You will understand as we proceed with the mechanism of this. First and foremost, that one definition that everyone remembers by heart, right from the day as they enter medical school till the rest of their life, is that apoptosis is programmed cell death. Apoptosis is programmed cell death. Right? Now cell death everyone understands, we've already discussed, but we've added a term over here, programmed. Now what is the meaning of this programmed? Means there are certain genes that are regulating this apoptosis. This is going to be regulated by certain genes. Now students, this is that one list which most of the medical students tend to fear the most. Something that you fear, the examiners will give maximally in the exam. So let's make a simplified version of this list today. What are the genes that are going to control apoptosis? So we have divided them into three categories. The first category is the sensors. Now whenever the body has any injury, any insult, the body has cells and cells have sensors which are going to sense that insult. What are those sensors? These include BIM, BID, BAD. So some similar sounding rhyming words, BIM, BID, BAD. Along with that we have Noxa and Puma. So remember these, all of these are the sensors, BIM, BID, BAD, Noxa and Puma. The second one that we have, the second list of genes that we need to know, genes which increase apoptosis. What do we call them? We call them pro-apoptotic genes. So they come under the category of pro-apoptotic. What are the genes? What are the list that we need to know under this? This will include the first and the most important, that is P53. First and most important. Other than that, it includes BAC, something similar sounding. On the C, add an inverted C, so BACs. So we had BAC, then we had BACs, and now we have BCLXS. So I told you some really tough words coming your way. P53, BAC, BACs and BCLXS will be pro-apoptotic. If you've done pro, you will also do anti. So that becomes our third in the list. The third one that we have which regulates apoptosis is anti-apoptotic. What do we have in this particular list? We have BCL2, MCL1 and BCLXL. Now very commonly the question comes, which of these is pro-apoptotic, which of these is anti-apoptotic? This list is confusing, I agree. How can we simplify this for our learning purpose? Now just focus on the anti-apoptotic category. BCL2 has L, MCL1 has L, BCLXL has these two L's. So how you can remember this is everything in the option that will have the letter L will be anti-apoptotic. Everything will be anti-apoptotic which has the letter L. BCL2, MCL1 and BCLXL. Now which of these other proteins also has L? That is this one, BCLXS, but this is coming under the pro-apoptotic category. So how will you avoid this confusion in the exam? One is BCLXL and one is BCLXL. BCLXS, S means stimulate. So stimulate means it will be pro-apoptotic. L means lower. L means lower, so it will be anti-apoptotic. So with this way of mugging up, I'm sure this list will become slightly simpler for you. Also, one question from this list only that will come up. Which out of these is both pro-apoptotic and anti-apoptotic? Now you can see there is one molecule that is coming in both the lists, BCl. For pro-apoptotic it is BCl-XS, for anti-apoptotic it is BCl-XL. So I can say that the entire BCl family, if I broadly say entire BCl family, this can be both pro-apoptotic also as well as anti-apoptotic also. I would be correct in saying that the BCl family has properties of both. Now students, remember, this particular list is one of the favorites with the examiners, a very important list that we have. And with this particular mnemonic that everything with L will be anti-apoptotic, I'm sure the options will now appear simpler to you. Since we have discussed this category of list, let's quickly finish one MCQ about BCl2. Now BCl2 is something that is present on cells of our body known as memory B cells. In fact, they are saved because of this BCl2. Memory B cell, from the name B cell helping us in our immunity. Memory, it is supposed to memorize that yes, ये infection पहले भी body में आई थी, ये चीज़ कौन memorize करता है, ये B cell memorize करता है, memory B cell, ताकि next time the infection that comes, the cell remembers it. So what are we going to see? Do we want such a cell to die? No. Do we want such a cell to undergo apoptosis? No. Means we want it to come under the mechanism of anti-apoptotic. Some anti-apoptotic protein will protect it from apoptosis, and that anti-apoptotic protein is BCl2. So BCl2 is then the apoptosis of memory B cell is prevented. It is prevented by BCl2. This is one very important, a newer question that is coming up, and what I want to add on to this knowledge. When a question comes once in the exam, you should try and read something about it more in detail so that the future exams might have something in that particular topic ahead. BCl2 activity on the memory B cells, this BCl2 on the memory B cells is going to be regulated by nerve-derived growth factor. Very important. Nerve-derived growth factor. So nerve-derived growth factor will regulate BCl2. BCl2 will then help in preventing the apoptosis of memory B cells. Within this list, there is one more important MCQ that is present about MCL1. MCL has L, so this is anti-apoptotic. Now we know that for any cancer, the most common treatment that is given is chemotherapy. But there are some patients who might not respond to chemotherapy. What is chemotherapy doing? Killing the tumor cells. I can say apoptosis of tumor cells, correct? But if there is a person who is not responding to chemotherapy, or the tumor cells are not dying in response to chemotherapy, means that there is some anti-apoptotic protein that is preventing the apoptosis. That is the next question that we need to know. MCL1 is most commonly associated with chemotherapy resistance. It is most commonly associated with chemotherapy resistance. So a person not responding to chemotherapy, which anti-apoptotic protein should come to your mind? MCL1. Memory B cells not dying in our body, not undergoing apoptosis, which anti-apoptotic protein should come to your mind? BCL2. So with this, we finished the main list of the proteins that are going to help us in calling apoptosis as programmed cell death. Now this is going more into the mechanism. Let's discuss a few basic points about apoptosis, then we'll proceed with the actual mechanism of apoptosis. The first thing about apoptosis that you need to know. Remember when we did necrosis, what was the definition? Cell death associated with, you are right, inflammation. But apoptosis has a very important difference over here, there will be no inflammation. A very important question that has been asked repeatedly in the AIIMS exam. There will be no inflammation, never associated with inflammation. The next question. Remember that necrosis was always happening because of some disease. For example, caseous necrosis was because of which infection in our country very commonly? You are right, tuberculosis. Fat necrosis in the breast was happening because of, you know it is trauma. So wherever necrosis was happening, there was some disease or pathology that was carrying out necrosis. But apoptosis is different. Apoptosis can be both physiological. Apoptosis can be both physiological as well as it can be pathological. Physiological and pathological. So is apoptosis occurring normally also in our body? Your answer is yes. Now what are the examples? Examples of this condition are very important. Talking about physiological apoptosis, remember the first example which is a favorite with the examiners is embryogenesis. Indirectly I can also call it organogenesis. Right? Embryogenesis, when the body parts are being formed in the intrauterine life, that is when a lot of structures are being formed and they are differentiating into mature forms. That is where apoptosis is very important. For example, when there is the formation of the hand. Now in the hand, when one mass is formed which is going to ultimately form the hand, for the fingers to form, in between the tissue must undergo apoptosis. Only then the fingers will form. If the tissue remains in between the fingers, all the fingers will be joined together. If they are joined together, the patient will suffer from syndactyly. So a simple understanding of this concept, embryogenesis is definitely requiring apoptosis. Next example. Now whenever any cell has served its purpose, at the end of the purpose the body wants to kill that cell. For example, some particular bacteria enters the human body. After the bacteria enters the human body, what will happen to the total leukocyte count? You are right, it will increase because our body is wanting to fight against the infection. हमारी body उस infection से हमें protect करेगी. How? By increasing the total leukocyte count. After a certain number of days, when the body has eliminated that bacteria, over a few days, when the person starts going into recovery, now what will happen to the total leukocyte count? It will come back to normal. How did this happen? Those cells which had increased, where did they go? They then underwent apoptosis. So that is the second example. Cells which have served their purpose in our body, they will be eliminated by apoptosis. The third example that we need to know, any harmful cell in the body, For example, if we have self-reactive antibodies, if there is something in our body that is going to come and kill our own cells, so if there is any self-reactive cell or antibody, any condition in which there are self-reactive cells, those are going to get eliminated by apoptosis. So these number of examples are definitely enough to understand what is happening in apoptosis. Remember, apoptosis can be normal. It is physiological also. The most important example that you need to know over here will be embryogenesis. Remember, this is one of the most favorite with the examiners. Now moving on to pathological apoptosis. Which disease can result in apoptosis? So there is one condition that we were just discussing when the patient is given chemotherapy. So cancer cell death. When a patient of cancer is given chemotherapy, how do the cells die? They die by apoptosis. Now what we must know, actually when the cancer cells die, it is a combination of apoptosis as well as necrosis. Actually, a combination of both, but most of the questions will have these as separate options. Then remember, it is more of apoptosis than necrosis. So again, one of those questions which you need to read very carefully. If you have an option of both, then that is your best answer, you are safe. But if you have both as separate options, then apoptosis will be the better answer for this case. That is the first pathological apoptosis, chemotherapy. The second one is what we see in viral hepatitis. In viral hepatitis, there is formation of apoptotic bodies. Formation of apoptotic bodies. But in viral hepatitis, the same apoptotic bodies have been given a special name that is known as the Council-Man bodies. These are known as the Council-Man bodies. So apoptotic bodies are Council-Man bodies. Which condition? In the liver, that is viral hepatitis. The last example in this list, friends, that we need to know is an example that I've taken from ophthalmology. Everyone has heard of a condition called retinitis pigmentosa. Retinitis pigmentosa. What exactly is retinitis pigmentosa? It is a condition in which the person has difficulty in seeing in dim light, or night blindness you can say. But the basic of this lies in apoptosis. What is happening? Everyone knows we have primarily two things for vision. One are the rods, another are the cones. The rods are going to help us in viewing in dim light, and the cones are going to help us in viewing in sunlight or daylight. So rods will help us in dim light, cones will help us in the, with the O you can remember the sun, that is the daylight. In retinitis pigmentosa, what happens? Selectively the rods undergo apoptosis. There is apoptosis of the rods. Now if there are no rods present, rods are undergoing apoptosis, what will this person clinically have? There is night blindness. There is difficulty in seeing in dim light, that is night blindness. So students, these happen to be the list of diseases that you need to know, pathologies that you need to know, which can result in apoptosis. Now if you have to select the most important ones, remember, for physiological apoptosis, always remember embryogenesis. For pathological apoptosis, a favorite with the examiners is viral hepatitis. What do you call an apoptotic body in viral hepatitis? It is also known as Council-Man body. So students, we have discussed that apoptosis is a type of a programmed cell death, which is then programmed because many genes are regulating it. It will not have inflammation, never ever will it have inflammation. It can be both physiological and pathological. Now we come to a more complex part of apoptosis, that is the mechanism. A very complex but again a very important part, mechanism. Before we start with the mechanism, there is one thing that you need to know. In the mechanism, we will talk about something known as caspases. Caspases. And I am now going to just split this term so that we understand. Now I will write it as this. What is this C? C stands for cysteine. So these are going to contain cysteine residues. All of us know whenever at the end of a particular word we add the word ase, we are dealing with some enzyme. So caspases are nothing but enzymes. They are going to break something. Enzymes always break something. Where do they break? They are going to break after the ASP for aspartic acid residue. For aspartic acid residue. So caspases are nothing but enzymes containing cysteine residues which are going to break after the aspartic acid. That is why we are calling them caspases. Now caspases are of two categories. Very important. If you remember these numbers, half of our apoptosis mechanism is done. These two numbers include two categories. One category is known as the initiator caspases and the other category is known as the execution caspases. What are the numbers for initiators? Remember 8, 9, 10. Caspases 8, 9 and 10. In order 8, 9 and 10. What for execution? For execution we have 3, 6 and 7. In this list now there are one or two more MCQs that are important. 8, 9 and 10 I am saying are initiators, but out of these caspase 8, it is not associated in apoptosis in humans. In humans, apoptosis will not include caspase 8, it will include 9 and 10. So humans are higher species, they will include the higher numbers, they will not include the lower number, that is 8. 8 will be involved in microbes, the lower species. Out of the execution caspases, what question do you get? Out of 3, 6 and 7, which is the most important? So remember, caspase 3 is the most important out of these. So friends, we've done the main enzyme that is responsible for apoptosis, that is caspase. Now with this we are going to begin with the pathways. Remember, do keep this in mind, the numbers of caspases, because the pathways will become very simple if you remember these. So what are the pathways for apoptosis that we need to know? First, let's make a broad outline and then we'll go into the details. Now friends, just like for caspases we did, they can be initiators and executors. So apoptosis is divided into two broad categories. One is known as initiation. One is known as initiation and the other is known as execution. Remember, initiation is the more complex one. If you get initiation, if you understand that once, execution is just a point or two. Initiation is further divided into two categories. That is the extrinsic pathway, and the other becomes the intrinsic pathway. So initiation and execution. Initiation can be extrinsic or by intrinsic pathway. The same order, let's begin the discussion of apoptosis. First we will start with initiation. Under initiation we will first start with the extrinsic pathway. So moving on, starting with the extrinsic pathway. Now extrinsic, what do you understand? Something outside. External means extrinsic. So what outside? So for example, this is a cell that has to undergo apoptosis. This is the cell which has to undergo apoptosis. On the surface of this cell, there will be something known as CD95. There is something known as CD95. CD95, my friends, is also known as FAS. It is also known as FAS. Now, if I say that the apoptotic cell has CD95 or FAS, there will be another cell with which it will interact. Extrinsic means outside, two cells have to interact. There is a cell that is the T cell. And this T cell is going to contain the ligand. So, this T cell will then contain the CD95 ligand, or I can say the FAS ligand, So one cell has the ligand, other cell has the receptor, both of these are going to combine. Other way round, other than this combination, there is one more combination that can come up. This can be binding between TNF receptor and TNF alpha. So either we have CD95 and 95 ligand binding, or we have TNF receptor and TNF alpha binding. As soon as this binding takes place between CD95 and 95 ligand, what happens next is that these three are going to associate. So I can say that CD95 trimer is formed, or trimerization is going to occur. Three CD95 molecules are going to come together and bind with each other, forming a trimer. As soon as this happens, what is recruited is a special molecule known as FADD. What is it attaching to? Fas. So what will I call it? It is known as the Fas associated death domain. Fas associated death domain. So because it is associating with Fas, I'll call it Fas associated, and this is going to ultimately result in apoptosis, so I'll call it a death domain, right? So where did we start from? CD95 ligand and 95 will combine such that three molecules of CD95 will come together to form a trimer. They will recruit a molecule called Fas associated death domain, and this in turn will then activate the caspase. Remember, caspases are present in an inactive state known as procaspase. So procaspase 8 and 10 will get activated into caspase 8 and 10. Now students, we were doing initiation. What were the three initiation caspases? 8, 9 and 10. Out of these three, which two have we activated? 8 and 10. Out of these, which will not be present in apoptosis of humans? That will be caspase. You are right, it is 8. Now till now we've activated 8 and 10. What are we left with in the initiation? Activation of caspase 9. Before we go to that, one point which will finish off this extrinsic pathway is, just now we said that this procaspase is converting into caspase 8 and 10. This pathway can be inhibited. This conversion can be inhibited by something known as flip. So flip, question that comes, that has now become a recent favorite, is flip anti-apoptotic or pro-apoptotic? So you know, flip is preventing the conversion of caspase, means flip is anti-apoptotic. Flip is anti-apoptotic. So with this students, we have done the first pathway, that is the extrinsic pathway. Now we'll go on to the next pathway, that is the intrinsic pathway. The next pathway is the intrinsic pathway. What I want you to recall over here is that we are discussing initiation. Initiation has two pathways, extrinsic and intrinsic. Extrinsic will have the use of CD95. Intrinsic is what we have come to right now. Now intrinsic means something inside the cell. So for example, this was the cell. Every cell for its growth needs a growth factor. There are some cells in the body which will need certain hormones. For example, the endometrium needs estrogen. So you can say hormones. So a cell might need a growth factor, a cell might need a hormone. But when the growth factor levels are going to decrease or the hormone withdrawal occurs, then what happens in the cell? This change is then going to be sensed by certain sensors in the cell. This change, that it is not getting its growth factor, it is not getting its hormone, is sensed by certain sensors. The first thing that we discussed in apoptosis, what are the sensors? Please recall that list. The sensors were BIM, BID, BAD, and you know it is NOXA and PUMA. Now these are the sensors which will sense this particular thing. What will they do? They will then go and activate. They will activate the pro-apoptotic factors so that apoptosis increases. And what will they do? They will inhibit the anti-apoptotic factors. So again, I want you to recall the list. Which were the pro-apoptotic factors? Remember, we had done the pro-apoptotic factors included, most important, P53. You are right, BAX, and BCLXS. That is how we had remembered, S for stimulation. What were the anti-apoptotic factors? All of us know everything that had L, BCL2, MCL1 and BCLXL. Now what have the sensors done? They have activated the pro-apoptotic factors. They have inhibited the anti-apoptotic factors. What will this result in? The balance has now shifted towards apoptosis. So because of this change that has occurred, one very important feature happens in the favorite organelle of the chapter. Remember, we had discussed right in the beginning, the favorite organelle for today will be the mitochondria. So a very favorite thing that will happen in our favorite organelle, the most important step, students, remember, the most important step is that there is going to be release of cytochrome C from the mitochondria outside. Cytochrome C. A very important factor. Cytochrome C, till the time it is present in the mitochondria, you know from biochemistry, it is helping in oxidative phosphorylation. That will help in generation of, you know it is ATP. But now because of the pro-apoptotic factors increasing, this cytochrome C is going to come out of the mitochondria. Along with cytochrome C, two more molecules are going to come out, and these include SMAC and Diablo. SMAC and Diablo are also going to come out. Remember, SMAC, Diablo, cytochrome C. All of them have come out of the mitochondria with a purpose. What is that purpose? Our end motive is to achieve apoptosis. So will they be pro-apoptotic or anti-apoptotic? All of them are going to be pro-apoptotic. All of them will be pro-apoptotic. Most important question comes on cytochrome C. How is cytochrome C causing this apoptosis? Cytochrome C then combines with something known as APAF1. Full form? If you know the full form, you will know what are we talking about. AP for apoptosis, AP, A for activating, and F for factor. Apoptosis, activating factor. So obviously we are moving towards apoptosis. The most important question of this pathway is this combination. Cytochrome C combines with which molecule? Straight one-liner question that you can't get wrong now. Cytochrome C combines with APAF1. And this combination, friends, this combination is now known as an apoptosome. So another question you've done. Apoptosome has which two things? First, cytochrome C. Second, apoptosis activating factor 1. Now there are some questions which might not be of any clinical relevance, but if they are asked previously, we must know them. Apoptosome has a structure that is, it is a wheel-like hexamer. Now this has been asked previously, hence we must know it, although it might not be useful in our clinical practice like other points, but this is what we must know. Now finally, if you see, we were discussing the intrinsic pathway. All the sensors were activated, pro-apoptotic factors were activated, cytochrome C has come out, it has combined with APAF1, formed an apoptosome. Why is all of this happening? What was the caspase that we were left with for activation? You are right. We had activated 8 and 10. We were left with caspase number 9. So this apoptosome formation is mainly being done because this will then convert pro-caspase 9 into caspase 9. So we've achieved the initiation. Going back, if I just want to recall quickly, initiation had two pathways, the extrinsic pathway and the intrinsic pathway. Extrinsic pathway activated which two caspases for us? 8 and 10. Intrinsic pathway activated caspase 9. You are right. So finally, we have activated 8, 9 and 10. Which stage will we now enter into? Go back to the first flowchart. First was initiation and then we had to go into execution. What were the caspases for execution? Yes, 3, 6 and 7. Which was the most common one? The most important one? That was caspase 3. So once caspase 8, 9 and 10 enter into execution, they are going to activate caspases of execution, that is 3, 6 and 7. Now friends, remember right in the beginning we did caspases have the word ase, they have an enzyme activity. What are they going to activate? These enzymes in turn in the body activate proteases to break the proteins, because it is a cell death happening, to break the proteins, and second, nucleases to break the nucleic acid. But remember over here we will use the term endonuclease. The most important, the nuclease that is activated in apoptosis is the endonuclease. Now briefly, we had initiation. Initiation had intrinsic and extrinsic pathway. 8, 9 and 10 were activated. Then we entered the execution pathway. Caspase 3, 6 and 7 were activated. They activated proteases and nucleases. What is happening now? Proteases will break the proteins. Endonuclease will break the nucleus. This we already know. How is that going to happen? So for example, if I draw that this is a cell and this is the nucleus of the cell. If this is the cell that has to undergo apoptosis, it is not that the entire cell will completely break down in one go. Remember the first line that we discussed. Apoptosis is a Greek word, means falling off. What is falling off? Bits and parts of these cells will break. Bits and parts of these cells, including the membrane, the organelles, the nucleus. So in small pieces, this is going to break off. So small pieces are falling off from the main cell. तभी हमने पढ़ा था apoptosis का Greek origin है जिसका मतलब होता है falling off. What is falling off? These small pieces are falling off. These falling off pieces are known as the apoptotic bodies. The next important question that we have, that is apoptotic bodies. Now students, remember apoptotic bodies, the question that you get, what are apoptotic bodies? So I can say a part of the cell membrane came out. Also with it, some parts of the organelles came out. Maybe the endoplasmic reticulum fell off, maybe the lysosome fell off, maybe a part of the nucleus fell off. So I can safely say this apoptotic body, they are nothing but cell membrane bound organelles. They are cell membrane bound organelles, a direct question from the previous board exams. What is an apoptotic body? Apoptotic body is nothing but a cell membrane bound organelle. Now we have gone too much into the mechanism. What we can't ignore, that we have to know the appearance under the microscope, without which pathology is incomplete. So when we are talking about an apoptotic body, how does it appear microscopically? The microscopic appearance is very important. So for this we will have to see one important image that we have, that is this particular image. Now if you focus on this image, there is one cell. In this entire image there is one cell which appears strikingly different. I hope everyone can visualize that it is this cell. What are the features of this cell versus the other cells in this diagram? And if you know this, you already know the features of an apoptotic cell. Now remember, if you can see, the overall size in this is smaller. Of this cell, the size is smaller. So I can say there is cellular shrinkage, cellular shrinkage. Second, if you focus on the nucleus, just focus on this round nucleus, the color of this nucleus appears light blue or dark blue in comparison to, say, this nucleus. Definitely it is darker in appearance. So this particular nucleus becomes the second point. Why is the nucleus appearing darker? Because all of the nuclear chromatin has condensed. If everything condenses into one body, the color becomes dark. So this is nuclear chromatin condensation. This is nuclear chromatin condensation. And remember friends, if we are put up a question that what is the hallmark of apoptosis, it would be this. A very important one-liner question. Nuclear chromatin condensation is a hallmark of apoptosis. Hallmark of apoptosis. A very important single line question. Remember first, every line that we have written in this particular slide is important. The first thing you need to know is this image. What is this image? This image is of an apoptotic cell. Second, what is the size? Is it smaller or larger? Remember, it is shrinkage. Now what you can recall, right in the beginning we discussed reversible cell injury. What was the first change in that? It was cellular swelling. It was hydropic change. So mostly all the cell deaths will start off with cellular swelling, except apoptosis, which will show cellular shrinkage. The size of the cell will become small. Very important high yield information for the exams that you need to know. Second question, what is the hallmark of apoptosis? It is nuclear chromatin condensation. Now since we have discussed the hallmark of apoptosis, there is one additional, you can call it a collateral question that we must do with this. If instead of putting up the question as hallmark of apoptosis, the examiner would have asked, what is the hallmark of neuronal apoptosis? So now you would say that apoptosis is occurring everywhere in the body. So what is so special about apoptosis in the neurons, neuronal apoptosis? So remember, neuronal apoptosis is different because neurons do not have caspases. The caspases are absent. Now if caspases are absent, friends, how is apoptosis happening? Apoptosis is happening because of apoptosis inducing factor. Apoptosis inducing factor. So remember, the hallmark of neuronal apoptosis is actually apoptosis inducing factor. The hallmark of just apoptosis in general becomes the condensation of the nuclear chromatin. These are two very important questions that come up, and not to forget the image that we have. This is of an apoptotic cell. Now if the examiner would have said that such an apoptotic cell is seen in the liver. We just discussed in the liver there is something known as viral hepatitis. Viral hepatitis, apoptotic cell, what would you call it? Correct, it is known as a Councilman body. Coming back to the theoretical aspect, apoptotic bodies have been formed. So for example, this is what I draw. This is an apoptotic body, and in the center there is a condensed nuclear chromatin or nucleus. Now students, apoptotic body has been formed, but will the body want to keep a dead cell? No. If a cell has died, our body will make sure that that cell is eliminated from the body. How is this eliminated? The apoptotic body is eliminated by a process that all of us know, that is phagocytosis. Phagocytosis. What you need to recall now, which is the cell that causes phagocytosis? Which is the cell that will come and eat this apoptotic cell? Phagocytosis is by macrophages. Phagocytosis is by macrophages. So a macrophage has to come and eat this apoptotic or this dead cell, and our body will get rid of it. Nothing can be as simple as it just sounded. There has to be some underlying mechanism, and a very important mechanism that we have over here. We know that every cell membrane is actually a lipid bilayer. This is a basic concept that we have. There are some proteins that are present inside the membrane on this aspect, and there will be certain proteins that will be present outside, that is on the outer leaf. Now what happens in an apoptotic body? There is one protein that moves from the inside to the outside, and that protein is phosphatidylserine. Phosphatidylserine. So there is a cell, apoptotic cell. Phosphatidylserine is a normal structure or a phospholipid that is present on the inner leaflet. In every cell, always present on the inner leaflet. But in an apoptotic body, it will come onto the outer leaflet. Why? Because once it comes onto the outer leaflet, this will then be recognized. This will be recognized by the macrophages. Once it is recognized by the macrophages, what will the macrophage do? It will undergo phagocytosis. It will eat up this apoptotic cell and our body will get rid of it. This is a concept that we have formed. The actual question that comes is one more point that you need to add. If I want to test this, that has phosphatidylserine come from inside to outside in this apoptotic body, what is the marker for this? For testing this, that phosphatidylserine has come out, there is a marker, and that marker is known as annexin 5. Remember, this is one very important question. Annexin 5 is what is testing the movement of this phosphatidylserine. There are two questions that you need to know over here. What is the marker of apoptosis? Correct, we just discussed. The marker is annexin 5. But if you don't read the question carefully, you might get this wrong. If the examiner asks us which is the molecular marker of apoptosis, the molecular marker. So again, the examiner has added one term. Which is the molecular marker? The first CD molecule that we had done. What was it? CD95. What is the other name for CD95? You are right, it is FAS. So please read the question carefully. Marker apoptosis का पूछे जाए तो it is annexin 5. Molecular marker पूछा गया है, then you will mark it as CD95 or FAS. So these are important things about how finally the apoptotic body will get eliminated from the body by a process of phagocytosis.